Hello, we're glad you've joined us for this live webinar, Isolation of Immune Cells Using StraightFrom MicroBeads Without Density Gradients. I am Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Miltenyi Biotec. Miltenyi Biotec is a global provider of products and services that advance biomedical research and cellular therapy. The company's innovative tools support research at every level, from basic research to translational research to clinical application. This integrated portfolio enables scientists and clinicians to obtain, analyze, and utilize the cell. Miltenyi Biotec's technologies cover techniques of sample preparation, cell isolation, cell sorting, flow cytometry, cell culture, molecular analysis, and preclinical imaging. Their more than 25 years of expertise spans research areas including immunology, stem cell biology, neuroscience, and cancer, and clinical research areas like hematology, graft engineering, and apheresis. To learn more about Miltenyi Biotec, visit www.miltenyibiotec.com. Let's get started. You can pose questions to the speakers during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen, labeled Ask a Question, and click on the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Answer a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process of obtaining your credits. I now present today's speakers, Zhong Yu, PhD, and Gregory Winkels, research scientist. Zhong joined Miltenyi Biotec in 2016 as Global Product Manager for the Cell Separation Reagents products. In this role, she manages the launch and marketing of new technologies in the cell separation portfolio. Prior to joining Miltenyi, Zhong worked for Promega Benelux as a technical service scientist and later as a product manager for the Cellular Analysis and Proteomics portfolio supporting the Benelux and Nordic branches. Since 2015, Zhong has been the Strategic Collaborations Manager to promote Promega's emerging technologies. Gregory Winkels received his diploma as engineer in biotechnology from the Aachen University of Applied Sciences in Germany. He did his thesis on characterization of blood dendritic cell antigens at Miltenyi Biotec, where he continued his work as a research scientist on myeloid cells and novel cell separation technology. Gregor has long-term experience in separation technologies, immunology, and antibody generation. As a group leader in R&D at Miltenyi, Gregor is responsible for myeloid cell separation products and also new technological developments such as the StraightFrom and the Max Express cell isolation product lines. Zhong Yu and Gregor Winkels will now begin their presentations.
Hello, my name is Zhong Yu, as said, Global Product Manager at Miltenyi Biotec for Cell Separation Reagents. In today's webinar, we would like to introduce our new StraightFrom MicroBeads for immune cell isolation without using density gradients. So in the first part of the presentation, I will introduce Miltenyi Biotec for those that are not familiar with us, and also our cell separation portfolio. Then my colleague Gregory Winkels, a group leader in our R&D department, who developed the StraightFrom MicroBead with his team, will go in depth with on our StraightFrom MicroBeads. Miltenyi Biotec is a privately owned company with its headquarters in Germany, with more than 2,000 employees worldwide, and with more than about 500 of them working in our R&D and engineering department, we try to advance to develop tools for both basic, translational, and clinical research. Our headquarters is in Germany, but we are represented worldwide in more than 28 countries directly, and the rest of the world is served by our distributors. We have over 11,000 products, and with those products, we can support you in your entire workflow for sample prep to cell separation and cell analysis in both the research and clinical area. So for those that are not familiar with our Max MicroBead technology, I would like to introduce first the basic principles. Our Max MicroBeads are around 50 nanometer size. They are super paramagnetic particles conjugated to highly specific antibodies. With these antibodies, we can target specific cell types. Together with the Max columns, which contain a matrix composed of ferromagnetic spheres, these target cells are then captured using the super paramagnetic particles. Together with the Max separators, the columns have a magnetic field that is amplified by these separators by 10,000-fold. So the target cells are captured in the columns and then separated from the untarget cells. After elution of the target cells, we have our clean isolated cells. This is a fast and gentle isolation method which can be done both manually and with our instruments. Just to give you an overview of our Max Cell Separation Reagents, we have different portfolios, and the separating strategy can be both based on different techniques. So first of all, we have our Max MicroBeads. This is based on the colors that I've just introduced. We utilize here nano-sized microbeads, and we have different types of microbeads, among others ultra pure and Realis. The starting material for these Max MicroBeads are single-cell suspensions, and this is a positive selection method. As a counterpart, we have our Max Cell Isolation Kits. These are also based on columns and nanosize, but this is an untouched isolation method which removes the unwanted cells. Now, in this webinar, we will talk about the StraightFrom microbeads. They are also based on columns and nano-sized microbeads. However, you don't need to pre-purify your cells. You can start immediately from blood products such as whole blood, Buffy Coats, and Leukopaks. As a counterpart, we also have our Max MicroBeads. They are based on column-free technologies and have micro-sized beads. So with this introduction to our company and our cell separation reagents, I would like to hand over the microphone to Gregor.
Thanks, Zhong, for the introduction. Hello also from my side. My name is Gregory Winkels. I'm working in the R&D department at the Miltenyi Biotec for more than 18 years now on the development of cell separation reagents, and I would like to introduce to you today a new portfolio of separation reagents we developed for the easy and fast direct cell isolation from human blood sources. But let me first start with a question. How do you isolate your cells of interest from whole blood, Buffy Coat, LRSC, or Leukopak? So most of the time, I'm quite sure you use density gradients to get your PBMCs and to then enrich for your cells of interest. But as you all know, density gradient cell separation protocol is an extremely time-consuming process. The transferred and diluted samples must be laid onto a gradient media before the centrifugation appears, and then the monolayer is transferred into a new vial. A platelet wash must be performed, and then the cells can be counted before the received PBMCs can be used. And this whole process takes usually more than 2 hours before you have the cells of interest in hand. So the density gradient centrifugation has additional disadvantages, and first and most obvious is the loss of white blood cells during the process. We just talked about the long protocol, but you not only have a long protocol, there's only a lot of hands-on time that you need to spend. And depending on the user you also see variations, and especially when you have older blood samples, for example, you not often get clear PBMC layers, the formation of aggregates appears, and sometimes you have uneven depletion of thrombocytes or granulocytes. So the discussion about how harmful FICOL is for cells, we don't want to start here, but another important point is that also the density gradient centrifugation is not very easily automatable. So our intention was to directly skip the density gradient centrifugation, that you directly are able to go straight from your source of blood to the positive separation of your cells of interest. And this without density gradients, without cell counting, and so on. So just short overview of the different cell sources. There are 4 main sources which differ in their concentration of PBMCs in blood. For whole blood, the volume for sure is variable. You have a low PBMC concentration. Buffy Coat normally comes in the volume of 30 to 80 ml, depending on where you get your samples from. Have a relatively high PBMC concentration and is a waste product after blood donation. Leukocyte reduction chambers are coming from platelet apheresis. Samples, for example, have the highest concentration of PBMCs at a very small and highly dense concentration, most of the time between 10 and 15 ml. And the last but not least, the leukopaks, where you have large number of PBMCs, usually 7 times 10 to the 9 cells, or even higher than 1 times 10 to the 10. And the volumes extremely vary depending on the vendor where you get your sample from. It can be from 80 to even higher than 200 milliliters. So let's have a look at the workflow for our StraightFrom cell separation from Leukopak. The volumes, as I said, are extremely depending on the vendor where you get your sample from. Like there are different vendors in the US, for example, where 1/2 of a Leukopak sometimes is within 70 milliliters, and when you choose another one, you have them in up to 200 ml. This must not be the case with regard to the cell concentration, but the total cell numbers are more or less mostly depending on the donor where you get the sample from. So what we did, we configured the StraightFrom leukapheresis reagents in a way that you can start with harmful leukapheresis, and you just need to adjust the volume of your apheresis to a total volume of 200 milliliters, and then you need to add the complete content of 1 vial of our MicroBeads to the sample. Then you can incubate your sample for 15 minutes with the MicroBeads, and then the separation is done using a multi-24 column block placed in the Multimax device. And this process usually only takes 30 minutes, so meaning without density gradient, without cell counting, no wash after labeling, you have your cells of interest within 30 minutes of time. So for sure, for the Leukopak separation, you would need the Multimax Cell 24 separator plus. This is a really great device when you're working with larger volumes or multiple samples, as we mostly do here. And in combination with the multi-24 column block, the user-friendly handling of large volumes is extremely comfortable, and the elution takes place with an automated vacuum station here. You can process half a Leukopak in a single run, and the device also comes with a single column adapter, which allows you to process also 1 to up to 12 samples in parallel with that, and this would be important for later use for the Buffy Coat and LRSCs when we are talking about this. In case you are looking for a fully automated high throughput system, we integrated the Multimax into a liquid handling system for high-speed processing, and this is the Multimax X device. Here you see now the separation of CD4 microbead from Leukopak. You see on the left side the flow data for before the separation, showing stained CD14 and CD4 markers, CD4 for the T helper cells, CD14 for monocytes, which also express at a low level CD4. And you see on the right hand, after separation, the highly pure T helper cell population. In the table below, you see the mean purity of this separation. This is a mean of 8 donors that has a very tight standard deviation, only with 2.7%, and the purity is really great with 95%. The recovery was nearly 70% as well, and the yield for sure depends mostly on the sample size and the donor frequency of the CD4 positive cells. An additional example here for StraightFrom Leukopak is the CD14 enrichment. You see here CD15 staining versus CD14, CD15 for the granulocytes, which might be problematic in case you have older samples, for example, and sometimes you see in co-enrichment, since CD14 is also expressed at a low level on granulocytes. But using our StraightFrom Leukopak reagents, you see here that after separation we only have really an extremely tiny amount of CD15 positive granulocytes left. The mean purity is again extremely great with higher than 94%, and the mean recovery with 56%, and the yield as before for the CD4 cells for sure simply is depending on the donor where you get your samples from. To sum the data for the Leukopak microbeads up, you see here the 4 different reagents we currently have. So we have CD4, CD8, CD14, and CD19, and you see there's a slight variation in the purity for CD8. This is depending on the number of NKT cells which also express CD8, and these cells we gated for the counting and the estimation of the purity was only the CD8 positive cytotoxic T cells. You see the recovery is really great. You have for CD4 and CD19 positive cells higher than 65–70% recoveries, and for CD14 and CD8 the recovery is good as well. This data here I also wanted to share with you, as these ones are about consecutive enrichment of more than one cell population out of one leukapheresis. So we all know how precious leukapheresis samples are, and therefore we wanted to get most out of it. And what we did here was the enrichment of CD8 positive cells, and we used the negative fraction before washing the columns for the subsequent enrichment of CD4. So meaning you simply take the negative fraction, label again, and do CD4 enrichment. And this works really well, as you can see here in the summary of the data analysis. You see that the purity of CD8 positive cells is, as before, 94%, but also for the CD4 cells enriched out of the negative fraction of the CD8 enrichment, you see that the purity is here good as well, and that you only have a minor contamination of CD8 positive cells in the pores. So, and this means that you can save precious material by doing at least 2 consecutive enrichments from one sample, what I think is extremely important. So, just short wrap up, using the StraightFrom Leukopak MicroBeads yields in excellent purities and recoveries, and it allows also for consecutive enrichment. So we currently have 4 markers available, and one kit. So what we offer contains all components needed for the isolation of cells from half a leukopak. So it contains the beads, it contains the multi-24 column block, and the whole blood elution buffer. And for the manual to full automation, we also provide the Multimax Cell 24 for the semi-automated separation in case you are interested in high throughput and full automation. We would also have the Multimax X. So meaning that within 30 minutes from even sizes like half a leukopak in a single run, you get your cells of interest out. Okay, now switch the blood sources and have a look at Buffy Coat. The volumes that are contained within a Buffy Coat are also a little bit depending on the transfusion center or blood bank where you get them from. They vary mostly between 30 to 80 ml. And what we decided was to make cell preparation most convenient and configure the beads in a way that you can use them without any cell counting, only by adjusting the content of your Buffy Coat with buffer to a volume of 80 ml. Then you simply take the content of one vial, MicroBeads, add them to your Buffy Coat suspension and incubate 15 minutes without any washing steps, and the cells can be applied onto the magnetic columns. We use here whole blood columns placed in the magnetic field of a magnet. And we need 12 columns for the enrichment of a complete Buffy Coat, and we apply there 7 milliliters per column. And as for the leukapheresis samples as well, here also we are done within 30 minutes without any density gradient centrifugation with the enrichment of your cells of interest. Very briefly, just a performance example. Here, again, CD14 from one Buffy Coat. You see staining again on the y-axis, CD14 on the x-axis, CD15 in this case. You see great purity with high recoveries, and even from the Buffy Coat you can get 2 times 10 to the 8 cells out in this case. As a sum up of our Buffy Coat reagents, what we have at the moment: CD4, CD8, CD14, CD19, and CD56. You see the purities are all above 90%, the recoveries and yields which are good, but for sure also depend a little bit on the frequency in the starting material. And as before, also here for the Buffy Coat reagents, we have possibility for consecutive separation. We developed at the moment markers for T helper cells, cytotoxic T cells, CD14 for the monocytes, for the B cells CD19, and for the NK cells CD56. And similar to how we configured also the kits for the leukapheresis, we here also provide one kit which includes all components for the isolation of cells from one entire Buffy Coat, meaning we have the StraightFrom Buffy Coat MicroBeads, we have the whole blood columns, and we have the elution buffers. And this separation you can do manual via the QuadroMarks or using MyDMAX, or what we prefer to use the Multimax Cell 24 or the Multimax X. Again, only 30 minutes for entire Buffy Coat on a single run. Let's come to leukocyte reduction chambers, which are more and more often used by researchers as an alternative source for white blood cells. And within one of these LRSCs, there are often more than 1 times 10 to the 9 white blood cells in 10 to 15 milliliters of buffer. And we elute these cells out of these LRSCs using syringes and buffer and fill the cell suspension up to 40 milliliters before we add then the content of 1 vial of our StraightFrom LRSC MicroBeads. Then with this, after this addition of the MicroBeads within 50 minutes magnetic labeling, you can take the samples directly and process them over the magnetic columns in the Multimax or in the QuadroMarks. Again, 30 minutes without density gradient centrifugation, no cell counting, no wash after labeling. And here just sum up of the data. You see that again, as good as the performance for the Buffy Coat, we have extremely high purity. The mean is always higher than 95%. The recovery is also extremely great. Lowest for CD56, this depending on the lower frequency of the cells in the starting material, and for sure also the yield here coming out of 1 complete LRSC. We are able to do also from LRSC consecutive positive isolations. We have the same markers as for Buffy Coat, and we are working for sure on more. And we configured the kit again as for the Buffy Coat together with the MicroBeads. This kit contains LS columns and the whole blood elution buffer. And as for Buffy Coat, you would need the QuadroMarks and the Multimax 24 or the Multimax X for the magnetic separation to get your cells of interest. So when we are talking about whole blood separation, the process here is as easy as for Buffy Coat and LRSC. What we do here is we simply add 50 microliters of MicroBeads per milliliter whole blood for the magnetic separation, and after 15 minutes again we go directly over 1 whole blood column, and for the direct enrichment of cells also again no cell counting, no washing is needed without density gradient centrifugation. The whole blood reagents were developed already a while ago, so the purities and recoveries are good as for the other reagents, but we have a larger number of markers available, so beside the markers for T helper cells, cytotoxic T cells, B cells, and then K cells, as well as monocytes, we also have markers for the enrichment of myeloid precursors like CD33 or for granulocytes like CD15 or CD66B, and plasma cells like CD138. For the manual to full automation, you can use whole blood columns, and you can use the QuadroMarks and the MiniMax for manual separation. Here we also have special protocols for our AutoMax Pro, the fully automated device, and you can use them also for sure on the Multimax 24 and via the Multimax X. So let's have a look at some applications. So first let's have a look at T cells, and what I want to show here is CD4 T helper cells, which were isolated directly from whole blood and then stimulated or not with IL-2 and T-cell activation and expansion kit. And what you see here is the expression of the maturation marker CD62-9. You see that without a stimuli on day 1, you don't see an increase. So there's no activation without any stimuli of the T cells, but you are able to stimulate them using the activation and expansion reagents to express CD69 at high level, and in the same level the expression of CD62L decreases upon activation. So what we also did here on the right hand, I showed the analysis of CD8 positive cytotoxic T cells, which were directly enriched from whole blood and also stimulated. And what we did here was we analyzed the secretion of interferon gamma and TNF alpha. So after 2 or 6 hours stimulation, and you can see that without any stimuli we don't have any cytokines secreted, so there's no unspecific activation of cells, and upon stimulation you see a high increase of cells which secrete the cytokines. CD14 positive monocytes is the next application I want to talk about. So ROS production is a hallmark of monocytes, and ROS production cannot only occur after stimulation, like TLR stimulation via LPS, or can also occur independent of stimulation. And this is something what you can see here. You see here confocal microscope pictures staining with cell rocks. This is a cell permeable fluorogenic probe. So what you see here on the left hand, you see the StraightFrom Buffy Coat, CD14 enriched monocytes, and you see no ROS production, whereas on the right hand you see monocytes which were separated via the reagents from a vendor, where you first need to do a FICO, so meaning you enrich your monocytes untouched but from a PBMC fraction, and you see here immediately that there's an increase in ROS production, and this is reflected here as well. You see on the right hand that the background of the ROS is extremely high, so that this lowers your assay sensitivity when we are talking about analysis of ROS production from monocytes. Let's have a look at the activation markers expressed on B cells, and what you see here are B cells enriched directly from Buffy Coat using CD19 and stimulated then for 2 days plus minus CpG. And you see here, as we already had for the T cells, expression of CD69, but also another marker of activation for B cells, CD86. And you see that without stimulation of CpG of the B cells, you see no increase on these activation markers, whereas after the stimulation you have a high increase of this marker. What we also analyzed via ELISA was the analysis of immunoglobulin secretion of B cells, and what you see here is the secretion of IgG, IgA, and IgM into the media after 7 days, so 5 days pre-incubation, and then stimulation with the CpG. And you see also that without any stimulation, there's no immunoglobulin concentration within the media that can be detected. So, last slide, I want to show you also again for B cells is Plasmablast differentiation, and what you can see here again is CD19 positive cells were isolated directly from Buffy Coat. The flow analysis was done for the Plasmablast to analyze via expression of CD27 and CD38. So in the Plasmablast differentiation was done with a cocktail of CD40 ligands, CpG, IL-2, IL-10, and IL-15, and then followed after a 3-day culture again with further cocktail, and plasma differentiation was then done with IL-6, 15, and interferon alpha. And so without any stimuli, you don't see any plasmablast differentiation. And this brings me directly to the summary of our talk. So you see that with the StraightFrom reagents, we developed a simple and fast isolation. It's possible from different starting materials as whole blood, Buffy Coat, LRSC, or Leukopak. There's no need to do any density gradient before you enrich your cells for interest. What's also extremely important when you talk to the people in the lab, that you don't need to adjust anything with regard to the cell numbers, so there's no need to do cell counting before, and we are extremely fast with our separation, meaning within 30 minutes you have your cells of interest. When we're talking about precious samples, you can also go for multiple subsets from a single sample, meaning you can do consecutive separations, and using the Multimax, this completes the package for your semi-automated cell isolation process. So, and with that, I thank you for your attention. I'm happy to take any questions.
Thank you, Zhong and Gregor, for your presentations. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question, and click on the Send button. Zhong Yu and Gregor Winkels will answer as many questions as time permits. The first question is, the half-sized leukopaks I usually receive are only around 100 milliliters. How should I use them?
This is rather easy. You simply take your sample and fill it up to 200 ml. Since we configured the kit that you can simply fill your sample to 200 milliliters, then add the MicroBead, the content of one vial, and incubate 15 minutes, and directly can go to enrich your cells of interest.
Is the Buffy Coat kit scalable?
Yes, that's also possible. You can process also half or quarter of the Buffy. When you use only half a quarter, the volume of the StraightFrom MicroBeads and the number of the columns you need to decrease accordingly.
Okay, thank you. Next one is, up to how many subpopulations can you isolate from one single sample?
So what we've tried so far was to go for 3 populations. What we tested was to starting with CD14 and then CD8 and CD4. But I think it would be also worth to give it a try to do more. We haven't tested it, but in principle it should work.
Thank you. Your next question is, what other markers do you have available?
At the moment we have the ones that we just showed. We are working on a few more markers, but in case you have specific needs, you can also reach out to us, and we would love to hear your specific marker wishes.
It looks like we have time for one more question. It will be, I am interested in testing these beads. Do you have samples available?
So, Paz, I can answer that question. We don't have specific samples available because we think this kit is shown the best when it can be tested in its whole, using, for example, a complete or a half Buffy Coat or Leukopak. So in that case, I would say reach out to your local Miltenyi representative or contact us, and we can see if we can have a demo at your place with your specific starting material and marker.
I would like to once again thank Zhong Yu and Gregory Winkels for their presentations. I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speakers via the contact information you provided at the time of registration. We would like to thank our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. This webcast can be viewed on demand through December 2018. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.